MIT alumni and friends. Welcome to tonight's discussion on one of the hot topics of the moment, the human microbiome. The work you'll hear about today can only be described as revolutionary, and that's not a term I use lightly. I'm Whitney Esbick, CEO of the MIT Alumni Association, and if you or someone you know is living with Parkinson's or an autoimmune disorder or a metabolic disorder like type 2 diabetes or obesity, or if you've ever taken an antibiotic and then considered taking an over-the-counter pro or prebiotic, you're going to be thrilled that you're joining us today. I'm excited for you to meet our guest speakers. You can read about each of them on the page below the viewing screen, but the quick version is this. Their unique partnership is completely altering what we know about medical science and human health. Professor Eric Alm co-leads the MIT Center for Microbiome Informatics and Therapeutics. His work saves lives. It's not hyperbole to say that this work will one day change each of our lives. He's joined by loyal MIT alumnus and corporation member Neil Rasmussen. You will hear Neil describe his own experience with an all too common immune disorder and how he formed a personal mission to accelerate the work of Eric's lab as a result. The research ecosystem they have partnered to build is revealing a new paradigm for understanding how our body works and how to treat it when it doesn't. And yes, because I know you'll ask, you'll all get a recording to share with your friends and family. We want you to spread the word. You will also get your questions answered today. We're fortunate to have Kelly Cervic with MIT's Night Science Journalism program as our moderator. Kelly writes for Science Magazine and is part of the MIT community this year as a fellow. She will moderate as well as work to weave in your questions throughout the program. So don't wait to add your questions in the chat box below. You can also up and down vote questions so Kelly can know what is of most interest to each of you. Thank you for spending time with MIT today. I'm going to stop talking now so that our guests can start. Here's Kelly Cervic.
Thank you, Whitney, and hello, everyone. It's great to be with you. Before I bring in today's speakers, I just want to emphasize the goal tonight is to make sure that you leave here knowing what the human microbiome really is, why it is the focus of so much cutting-edge science, and also how this MIT Research Center with its engineering and mathematics approach is contributing to those new discoveries. So this discussion is not going to be theoretical. We're going to be talking about real-world applications of microbiome science. And this is a really vast topic, and I know it's going to spark a lot of questions. So like Whitney said in the introduction, we're not doing a formal Q&A session at the end. I'm going to be trying my very best to weave your questions into the conversation. So please don't wait until the end. If there's something that you're curious about now, or you hear something that sparks another question, go ahead and type it in. So with that, I want to introduce the co-director of the MIT Center for Microbiome Informatics and Therapeutics, Eric Alm. Hey, Eric.
Good to see you, Kelly.
Yeah, welcome. So I'm sure many people tuning in have some notion of the microbiome, but just to make sure that we're kind of all setting out on this discussion from the same starting point, what is the human microbiome?
Good question. So I think we have a slide that shows some of the components of the human microbiome. And we can think of the microbiome as an ecosystem. It's the ecosystem of our body. So it includes thousands of different bacteria. And these bacteria live in our gut. They live on skin. They live in the vagina. They live in the nose, in the mouth. Really any part of the human body that is considered to be the external side. And besides bacteria, there's also eukaryotes. So there's fungi. There's animals. There's little insects living on our skin. There's viruses, and these could be human viruses that we're familiar with that cause disease. But there's also a very large number of what we call phage, and those are viruses that infect the bacteria that live on us. And we don't really understand how all of these, what their different functions are, and even some of the major groups of bacteria. We don't know what different roles they play. So we're really at the beginning of understanding the human microbiome. What we do know is that there are about as many bacterial cells in your body as there are human cells. And there are about 100 times more genes in those bacteria than there are in the human genome. So we know they impact gut health, but they impact the health throughout health and well-being of our bodies, well beyond the manifestations that we see in the gut. And everyone's microbiome is different. So I have a slide that I'm really happy to share with you guys, and it's a view of my microbiome. So in 2009, myself and my graduate student, Lawrence David, who's now a faculty member at Duke University, we tracked our microbiome every day for the whole year. And on top, you can see mine, and on the bottom is his. And if you look at it, you can see, there's the different colors are different bacteria, of course. And you can see not only do we have different species of bacteria and different amounts of each, but those bacteria are changing day to day. And so the microbiome, it's not a static thing like the genome. It's something that is constantly, changing and responding to the environment. What we know now is the microbiome is involved in a lot of different diseases. So everything from infectious disease, which is when a microbe kind of grows out of control, to metabolic disease like diabetes, obesity, autoimmune disorders like inflammatory bowel disease, MS, and things like autism, asthma, allergy, and more.
Yeah, so you're naming all of these different diseases. And, I think thinking about the microbes in our body as sort of a source of disease or disease risk, that is not how we've sort of classically thought about disease. I wonder why are most of us only recently learning about the role of the microbiome?
Yeah, when I started getting interested in the microbiome, it was maybe a little over 10 years ago. The first thing I did was to go to PubMed and look up all the papers, and I read every paper that was related to the microbiome, and it took me a few days. Today, there are more papers published probably every day on the microbiome than there were, in the entire time up to, maybe 2010 or something like that when I got involved. So why are we just learning about the microbiome? Is it, did we invent a new microscope, or, what was the spark? It was the human genome, because we made DNA sequencing really cheap and really accessible and really fast. And that's the best way to study this hidden microscopic world. So, we take a sample of it and then we sequence the DNA, and the DNA tells us which microbes are where. So DNA is kind of our microscope. And we've built a lot of tools that allow us to take these molecular measurements like DNA sequencing and special techniques that help us figure out, how many of each species is where. And that's part of what we do here at CMIT, the Center for Microbiome Informatics and Therapeutics, is we put those resources out there so other people can participate in microbiome science. Mm-hmm. And so CMIT is very generously founded and funded by the Rasmussen Family Foundation. And Neil Rasmussen is here with us today to have a discussion.
Yeah, let's bring Neil into the discussion at this point. Hi, Neil.
Hi, Kelly.
Hi, so Neil is not only a funder of CMIT, but he's also very passionate about this field and very involved in the lab. So maybe first, Neil, could you just sort of tell us about some of the recent research findings around the microbiome that have you most excited?
As Eric mentioned, the microbiome impacts more than just the gut. And we will talk about gut health, but I never cease to be surprised by the relation of the microbiome to other medical conditions. I'd like to just list some of the more recent research findings. And let me emphasize, as Eric said, these are coming out quite quickly, and the findings I'm going to describe now are just within the last few months. Regarding Parkinson's, new evidence shows that Parkinson's starts with a microbiome problem in the gut and travels up the vagus nerve. Endometriosis, a finding that abnormal gut microbiome contributes to endometriosis. Fertility, certain bacteria in the vaginal microbiome promote fertility. Depression, people with depression have an excess of certain strains in their microbiomes, strains which are known to be involved with the synthesis of certain neurotransmitters. And obesity, microbiome changes caused by the Western diet lead to metabolic syndrome and obesity. And more interestingly, that an appropriate diet is protective. Cancer, a diet including certain starches is protective against certain cancers for those people genetically predisposed. Chronic vaginosis, researchers have created a vagina on a chip a tissue culture that'll allow them to explore the mechanisms of chronic vaginosis. Autism, maternal gut inflammation during pregnancy can lead to autism-like symptoms in mice. And Alzheimer's, manipulation of the gut microbiome reduces tau-mediated brain damage. And all this is just in the last few months. Why are these happening right now? As Eric said, it's been enabled by advances made in the research tools, which MIT and the Broad Institute have been contributed to so much.
Yeah, you can tell just by listening to Neil, I think that he's one of these rare philanthropists who is just so engaged in the science. I think half of the times I hear about a new paper, it's because Neil has emailed it to me. and that's really helped us get things going at CMIT. And I'm proud to say that we've been a part of that real explosion and new discoveries and research since we opened the doors in 2014. We've had hundreds of scientific publications and actually started dozens of clinical trials.
And I just want to chime in here and say that if anyone was feeling like all of those different applications that Neil named went by your ears very quickly, I am told that Eric and Neil will be providing a list of links to some of the research that gets mentioned this evening.
I felt that way, Kelly. Like half of those things were new to me.
Yeah. So those links will go out along with the recording of our discussion early next week. If you're one of those people that likes to swim around in the footnotes and get a little deeper into the research, that is coming. So I want to start bringing in some audience questions, and we do have a few already. So let me just see what we have here. One is about food, which I think, sort of Neil mentioned sort of the Western diet, and I think people are really interested in that. Someone asked, how much do fermented foods change the microbiome? That's something that we knew was coming, and I know Eric, who's drinking his kombucha over there.
I'm drinking my kombucha right here. So obviously,
about that.
Yeah, obviously I'm a believer in fermented foods. There's actually been some nice recent work out of Stanford that shows fermented foods in general really promote gut health and are pretty good for the microbiome. Fermented foods tend to produce these short chain fatty acids like acetate and things like that. You're familiar with that vinegar taste. That's actually what's supposed to be going on in your colon. Your colon is really it's a fermenter. So you put dietary fiber in and you get these short chain fatty acids out. In our modern diet, we don't really put a lot of dietary fiber into our colons, and that may be one of the reasons why these fermented foods are so good for us.
Yes. And I know we're going to try to get into a little bit more of sort of how we can manipulate our microbiomes and like the role of lifestyle choices as we go along in the discussion, but I knew that one was coming, so I thought I would throw it at you soon. Let's pivot a little bit, and I want to talk about kind of where this field might be going. How do you both think about the ways that the study of the microbiome is likely to impact medicine, and what might that look like? And maybe let's start with you, Neil.
Well, beside learning mechanism about some of these diseases and indications, I foresee microbiome-related therapeutics for chronic diseases in conditions where we haven't made much progress recently, like inflammatory bowel disease, arthritis, allergies, Parkinson's, diabetes type 1. I foresee a time when microbiome-related therapies are engineered to specific conditions, replacing drugs and especially broad-spectrum antibiotics. And I'm excited that these therapies may have the potential to be made at very low cost. So it's an exciting opportunity to break down the barriers to health equity in developing countries.
Neil, I want to ask you about that. That was just a really interesting point that you made about cost. And I'm wondering sort of what makes you say that? Like, how are you thinking about that? Are you thinking about costs relative to biologic drugs, or what, how do you think about that?
Well, of course, the FDA process itself remains a stiff barrier to low cost.
Yeah.
But outside of the U.S., they may not have some of those same barriers. And you can imagine, obviously, making yogurt is pretty low cost. And it's possible that some of these agents might be naturally derived from things we find in microbiomes in other communities around the world.
Mm-hmm.
So it's possible that they're simply made in the same way we make, you can go buy lactobacillus drugs in the store.
Yeah. I think it's interesting to point out that maybe microbiome drugs could look a bunch of different ways, right? Like some of them could be sort of off the shelf, things that we know are healthful. Some of them could be more personalized.
Some of them may not be things we eat. They may be like nasal sprays or something like that.
Mm-hmm. Was there anything else you wanted to add about that, Eric?
Yeah, actually, I have a slide on how I kind of see microbiome drug development. And, traditionally drug development it's a very expensive process. And a lot of that process is identifying lead compounds by screening millions of compounds and then modifying them and looking for toxicity and things like this. In microbiome, with microbiome-based therapeutics, the ideal is that we can really take advantage of DNA sequencing. And so we start with a person, we sequence the DNA of their microbiome, we identify the bacteria, and then we can look and we can compare healthy people and sick people. And if we see that everyone who's sick is missing 1 or 2 or 3 bacteria, then the other thing we're doing when we take those samples of bacteria is we culture them. And so we take the bacteria, we isolate them in pure culture, and we put them in the freezers. So if we find there's a deficiency of bacteria, we can go to those freezer stocks, we can grow up as much of that bacteria as we want. And that's now a hypothesis for a clinical trial. If you're sick, you're missing this bacteria, well, let's grow it up. Let's put it in a pill. Let's have a clinical trial and let's see if it's efficacious. And that's there's a lot of companies out there right now that are thinking exactly along those lines. Like, how can we take advantage of what we already know about the microbiome to make these new microbiome-based therapeutics, these live biological agents?
Yeah, I think you sort of inadvertently spoke to another audience question. So I just want to say here, someone was asking, how can we measure our microbiome? You were sort of describing the process for isolating and sort of sequencing. And the second part of that question was, what makes a good microbiome? Which I think is a really sort of interesting question that, people think about it like, can we bin each species like, this is a good guy, this is a bad guy? And what you're describing is a little bit more like, let's look at the disease and see what people have in common. And then that might kind of point us to the bad guy. Do I sort of have that right?
I mean, that's the basic idea. I think at this point, nobody can really tell you who's the good guy and who's the bad guy. And so we're really at early stages. And sometimes if we don't see a certain bacteria in people who have a particular disease, does that mean that they have a disease because they don't have the bacteria? Or maybe they don't have the bacteria because they have the disease. So the causality is something that needs to be determined as well, right? So that's the basic idea, and we're kind of at the early stages, and nobody really knows what's good and what's bad at this point. Although there's certainly a few that we know are bad actors, right? Yeah. If you get a E. coli infection or something like that, that's obviously bad, but.
Right. Well, so thinking about good guys and bad guys some more, maybe that's a good way to pivot to probiotics, which I know, we received this question several times, even in advance of the program via emails. We're hearing a lot about probiotics. What are they? Should we be taking them? Where do we get them? Eric, I know that you were keen to tackle this issue a bit.
Yeah, absolutely. So I want to show you guys a slide, and this is a depiction, not a depiction, it's a photograph of 2 mice. And we, in collaboration with Dr. Susan Erdman at MIT's Department of Comparative Medicine, we ran this study where initially we accidentally fed mice yogurt. And then we repeated the study, just isolating this Lactobacillus reuteri, this probiotic bacteria that was in the yogurt. And what we found when we grew up those mice is that, so if you look at these two, these guys are brothers. They're from the same litter. They're genetically very similar. They live in identical conditions. They eat identical mouse chow. But you can see the one on the left is skinnier, it's leaner. It has, you probably can't tell from the picture, but it has actually shinier fur. So it has more sebaceous glands around its hair follicles, and its skin is healthier and thicker. And like amazingly, the wound healing happens a lot faster in the mouse on the left. And really this something that Susan calls the glow of health is conferred by these probiotics. And it just covers a whole list of phenotypes that really have nothing to do with gut health. Because we usually think about, oh, probiotics, it's good for your gut health. We're now starting to learn more about other effects around the body than we are in terms of effects in the gut. And in fact, there's not much Lactobacillus reuteri in the colon as a percentage of the bacteria that are down there.
Okay, so I want to be the shiny mouse. I want to have the glow of health.
Yeah, we all do.
I wonder, beyond probiotics, I think there's a big question of like, how much power do we really have with our lifestyle and our choices to influence that? And there is a lot of pseudoscience and a lot of sort of shaky advice out there in the popular press about what people should and shouldn't do. Eric, how do you think about what advice to take seriously and what to ignore?
Yeah, that's a really good question, Kelly, because when I get on Google, it prioritizes ads for me that have to do with microbiome, and I see the same ones that probably a lot of our viewers do. And there's companies that say, hey, we'll test your microbiome, which is totally legit, and can be a fun activity to figure out what's in your gut. But then they'll make personalized recommendations for you on what you should eat and drink and do in terms of lifestyle choices, things like that. Most of that, I would say, is pseudoscience. We can't translate those types of dietary changes to changes in the microbiome. We don't really know, what the healthiest microbiome is for any given person. And the other thing I hear a lot, I was just in a cafe the other day and somebody was saying, I got to get me a fecal transplant from a skinny person because I just can't take off the weight. And some of our viewers are probably saying, what's a fecal transplant? It's literally putting fecal matter from one person into another. And the idea that could make you skinnier or more athletic or any number of other factors is pretty much pseudoscience. What is science is a fecal transplant for certain diseases like recurrent C. difficile infection or inflammatory bowel disease. There's been a number of trials that show improvement in patients with IBD.
And I just want to say there's a lot of interest in the chat about fecal transplant and some questions about sort of why, like, we know it works for this and that. Why doesn't it work for the other? In particular, someone was asking about irritable bowel syndrome. What would you say about sort of what's been proven and what's not around fecal transplant?
Well, we don't know anything until we've done randomized controlled trials of a certain size. And so there have been a lot of trials in inflammatory bowel disease, and taken in aggregate, we're starting to see that, that they're, we're repeating this observation that patients are having some improvement. With irritable bowel syndrome and with a lot of diseases, it's probably not just one disease. And so that's what makes some of these trials really difficult. And so there could be a set of patients with irritable bowel syndrome that might be improved by a particular microbiome intervention, but maybe it needs a microbiome intervention plus a dietary intervention, and maybe it's only a subset of IBS patients. And so these are kind of some of the complications that make it difficult to see a positive effect.
Yeah, thank you. That kind of leads us back to a conversation about mechanisms and sort of like how is the microbiome working all these different effects and playing all these different roles in the body. And I wonder, Neil, maybe you can talk a little more about sort of how it is that microbes influence so many different illnesses and conditions.
Well, Kelly, I have kind of a simplistic explanation for why the microbiome is so critical to diseases related to the immune system. I think we can all understand how microbes can make nutrients that benefit us or make toxins that harm us. but many of the most interesting effects relate to the interaction with the immune system. We are all born with an immune system, but it grows and learns during our lifetime through its exposure to the microbial world. You could say the microbiome programs the immune system. And you can simplistically think of 3 ways that something can go wrong with your immune system in that regard. First, you can have an infection by something in the microbiome that the immune system is supposed to fix, but it can't. And it can linger as a chronic inflammation or illness. And we all understand the role of low-level inflammation in so many indications. We might be able to expose the immune system to microbiome therapeutics to better train it to handle such conditions. And second, the microbiome can program the immune system to improperly respond to something benign, like pollen or peanut butter. And that creates an allergy or chronic inflammation like sinusitis, as Eric mentioned. We clearly should be able to develop therapeutics to assist with that problem. And third, and this is really the most troubling, the microbiome can try and hide from the immune system by acting like human cells. And this could improperly trick the immune system to decide to attack your own cells, such as the so-called autoimmune diseases. Again, this is maybe a problem we can fix by training the immune system through microbiome-related interventions. In theory, it's possible to prevent and cure all of these types of problems through microbiome therapies that can help program the immune system. And it's the interaction with the immune system which is responsible for the broad range of indications and diseases, which, as we've seen, the microbiome affects.
Yeah.
We're actually starting to make some progress on this. If I could talk about some work that my co-director at the center, Dr. Ramnik Xavier, has been part of. I think we have a graphic for this. This is a really interesting story about. It's a city that is on the border of Russia and Finland, and after World War II, it was divided in half, and so there's really the same people, the same genetics, the same lifestyle, the same diet. But on the Finnish side of the border, hygiene levels rapidly increased after World War II. And on the Russian side, they stayed largely the same. And what they found is that the rates of asthma and allergy dramatically increased on the Finnish side, but not the Russian side. And what Dr. Xavier and his team was able to do is they went in and they looked at children on either side of the border, and they looked at what were the microbes they were experiencing early in life. They found that the kids on the Finnish side of the border, their guts were really populated with an abundance of a type of bacteria called Bacteroides, whereas on the Russian side, they were mostly populated with bifidobacteria. And they were able to identify compounds that were made by those Bacteroides that really interfered with the signaling that those children's immune cells were supposed to get from more pathogenic bacteria like E. coli and bacteria that were more abundant on the Russian side. And that may really contribute to these vastly different rates of asthma and allergy, on different sides of this city.
That is fascinating. I'm going to try to wrap into that discussion some other kind of exposure related questions that are coming up in the chat. One is asking you to asking about the potential for pesticides or antibiotics in the environment to kill microbes in soil and change the way that we are exposed to microbes from our environment. Is that something that you've thought about or something we should be worried about?
I don't know if it's something to worry about. To be honest, Kelly, I haven't thought a lot about that. There are, we are constantly exposed to antibiotics, and there are antibiotics that for a long time had been used in livestock. And part of the thinking was that, the bacteria that live in the guts of livestock, they don't really thrive in the human gut, so we have nothing to worry about. But we published a paper a number of years ago that shows that the antibiotic resistant genes, the genes that turn a bug into one of these super bugs, one of these antibiotic resistant bugs, they can jump from one species to another. And so, if we're ingesting these organisms that might not live in the human gut, well, the genes from those organisms can make it into the bacteria that do live in our gut. So I think it is important to be mindful about how we're using antibiotics.
Yeah, absolutely.
But I would like to comment on that. It's certainly scary to think about us manipulating the microbiome in the soil on any scale, but there's some reasons we might want to do it. For example, we have pollutants in the soil, like oil in the soil or pesticides. It's in theory possible to develop microbes that might try and like to eat those things. So it could be beneficial in that way.
That's absolutely right. I mean, there's a lot of projects that are handled through bioremediation, and that's, when it becomes too expensive sometimes to clean up a toxic site, we think about, well, what's the ecosystem there, and is there something we could do to stimulate that the microbes in that environment to clean up some of those toxins. We certainly saw that with the Gulf of Mexico spill. Most of that oil disappeared quite quickly. We actually went to, in collaboration with BP, we went to a number of different oceans around the world, and we conducted a similar experiment in test tubes, where we would put oil from that region into the seawater, and it turns out the Gulf of Mexico was I guess the best place to have the Gulf of Mexico spill, because the bacteria there were most able to quickly clean up that oil, and if it had happened anywhere else, it would have been an even bigger disaster.
Wow.
Eric, maybe this is a good time for you to just sort of talk more about MIT's role in the research field and sort of what you guys are doing over there.
Yeah, of course. I'm always happy to talk about that. So why are we doing it at MIT? Well, it sounds like a lot of microbiology, but when we got into this field, one of the first discoveries that we made was that we really need new mathematics to describe the dynamics of these communities. And so that's been a key feature of what we're doing. We've recruited some folks from the computer science department, like Professor Bonnie Berger, who's come up with ways to handle the enormous data sets that we're collecting. And we've been developing new algorithms that help us understand things like fecal transplants. I actually have a graphic here I'd love to show you about how that works. So I want you to just imagine that all of the columns on this graph, okay, are different bacteria. And so the darker the vertical line, the more of that bacteria there is. And if it's white, it means that bacteria is not present. So this is basically the microbiome profile for 3 different samples. The top one is a patient that's suffering from a Clostridium difficile infection before they get a fecal transplant to cure them. The second is the donor. This is the healthy person whose stool sample is being used to treat the patient on top. And on the bottom is what that patient looks like after getting the treatment from the donor. So what I'd love for our viewers to do for just like 10 seconds, look at the patient before transplant, look at the donor, and try to predict, like, what's the rule for predicting what the patient is going to look like after fecal transplant. So just look at that for a minute. So when I first saw this, and I think probably a lot of our viewers are thinking along the same lines. First, they said, well, maybe it's, A plus B, right? It's the top line plus the second one. And no, that can't be, because then it would just all be filled up. Or maybe it's an AND gate, or maybe it's an OR gate, or something like that. And we tried all those kind of simple explanations, and nothing seemed to work. So a graduate student who's now a professor at Mass General Hospital, Chris Smiley, came up with a machine learning algorithm that looked at a lot of different examples of these patients before and after fecal transplant. And let's show the next slide, because it shows how his algorithm works. And you can see it picks up most of the detail. So, even something as seemingly crude and empirical as a fecal transplant, we still need kind of, some of that MIT magic, some of those algorithms and new ways of approaching data to better understand it.
That's such a cool graphic. And something I find so fascinating about fecal transplant is that you're putting in like this amazing like smorgasbord of, we don't even know what came from, the individual donor stool. And so, yeah, it's going to be interesting to see sort of how that gets refined based on the data like you're collecting to figure out like, okay, well, what is the active ingredient? Like, do we have to use the poop every time, or can we, treat this a little more like a drug and standardize it? I wonder, Neil, if there was, I know you have had your mind on sort of the data problems around this field for a while. Anything you wanted to add about what's going on at MIT?
Well, there's hundreds of different kinds of microbes with basically an unlimited number of mutations, producing hundreds of different kinds of metabolites. The data that Eric gets from one person's microbiome on a single day can be 100 gigabytes. So this is really a big data problem, which is really one of the reasons why a lot of this kind of research, it's not for a medical school. It's really the reason why MIT is and has to be the leader in this field.
And I should say, it's not just about the data either. One of the things that we've been busy doing is collecting kind of the raw materials. So all of the individual bacteria, isolating them, characterizing them, putting them in the freezer, so that at some point we can move beyond some of these really crude tools like fecal transplant and broad spectrum antibiotics to like a precision editing of the microbiome, where we can do something like find a virus, find a phage that targets one particular bacteria, remove it from the microbiome, then go to the freezers and take a different bacteria and put it into the microbiome. So like really taking that MIT engineering approach to crafting the microbiome and engineering it. And then we can think about going beyond just changing what species are in the microbiome. We can figure out, what are the molecules that those species are making? Can we isolate those and make something that looks more like a drug? Or on the other side, we've got folks like Chris Voigt, Tim Liu, Jim Collins, who think about, can we engineer those bacteria to perform new functions that they didn't have initially, but that might be beneficial to our health?
Yeah, I want to chime in and just say that there was a question that kind of relates to what you're talking about, just asking about the mechanisms that we're even interested in here. They're saying like, do our gut microbes make enzymes, and that's what we're interested in, or are they digesting things that we otherwise wouldn't be able to? Is it all of the above? How do you think about sort of what's working for us?
Yeah, it's all of the above, of course. So what they're supposed to be doing, on the surface is they're supposed to be taking dietary fiber, fermenting that into something called short chain fatty acids. And that's what my kombucha here is full of. And in addition, a lot of those bacteria will produce other molecules. So, there's they modify tryptophan. So tryptophan, you've heard of, is the thing that makes you, amino acid that makes you sleepy and things like that. They'll modify tryptophan, and that signals specific receptors in the gut, and that's thought to be helpful for inflammation and a lot of different conditions. They make enzymes that affect our bile acids and can convert them into different forms. And so they do all of these kind of metabolic activities, but then they directly interact with the immune system. And so all of the things I told you about the probiotic, if we cut out the interaction between the bacteria and the T cells, you don't see any of those health benefits. Mm-hmm. And the other interesting thing is if you sever the vagal nerve, so the nerve that connects the GI tract back to the brain, you don't see any of those probiotic benefits. So they're really, they have multiple ways of sending these signals throughout the rest of the body.
Yeah, so not any one thing, really hard to define sort of the magic there. Talking about sort of harnessing those different effects and developing treatments. I know that there are several sort of microbiome related startups that have grown out of MIT's ecosystem. What would you highlight there, Eric?
Oh, there's so many. I was just talking to one of our former graduate students who's now CEO of a company called Concerto Biosciences, and they're taking some of the technologies that they developed as part of the Microbiome Center here at MIT and using that to both, develop their own products and help other companies develop new products. There's, probably a dozen microbiome therapeutic companies that are working on cocktails of organisms that might help alleviate various different diseases, like Series Therapeutics and companies like that. A grad student in my lab started a company that's sort of tangentially related to microbiome. And if you've heard at all about how they monitor the sewers, the wastewater in the sewers to see how much COVID there is, that was started by one of our graduate students at the center here. So all of those are kind of activities going on in the corporate world. And then, we've fostered an ecosystem here that's really friendly to nonprofits. And so the latest one, a couple of postdocs in my lab started a project called the Global Microbiome Conservancy. And one thing we know is that pretty much everybody in the modern industrialized world has basically the same complement of microbes, and these are really different from what we see in indigenous peoples living a more traditional lifestyle. And so as those populations have more access to our mass-produced foods and our antibiotics, we anticipate over the next 5 to 10 years that their natural biodiversity might start to look a lot more like ours, and we might lose these critical strains that have co-evolved with humans for millions and millions of years altogether. So we started an initiative to go around the world and collect those that valuable biodiversity and preserve it for future generations.
Someone asked,
and I'd like to bring up OpenBiome in this context.
Yeah,
it's a nonprofit therapeutics company that started out of Eric's lab, and it provides the gold standard treatment for antibiotic-resistant C. difficile using fecal transplants. They've cured over 65,000 people, many of whom had hours to live prior to treatment and walked out of the hospital the next day.
Yeah, there's some really remarkable stories around fecal microbiome transplant. I want to sort of weave in a question that came up thinking about sort of traveling around collecting microbiomes, preserving different types of microbiome. Someone asked, where do you go to find good microbiomes? Do you travel to places where people tend to live much longer? Do we borrow from other areas of the world? And how can we replicate that?
That's a great question. So one of the postdocs here at the Center for Microbiome Informatics and Therapeutics, he left and started his own laboratory at the Fred Hutch Center over by University of Washington in Seattle. And I know he recently published a paper on the differences in microbiome in some of these blue regions. And so folks are working on that, and there are different microbiomes there. One thing I would say is, our microbiomes probably don't act on their own. So, we look at some of these indigenous populations and they have very different microbiomes, but that microbiome might not work for you because they probably eat 10 times as much dietary fiber as you, and so part of it is figuring out what's a microbiome that's going to be compatible with your diet and lifestyle and things like that.
So you really can't sort of disentangle and just give someone a microbiome and expect it to work for them the way it works for someone else.
Right.
Very interesting. Okay, I know that you have a lot underway right now. So I wonder if we could pivot real quick and let you talk about sort of what things is MIT working on that are in progress that we should sort of keep an eye on.
All right, let's. I think we have a slide on one of my favorite projects right now, and that's called IBD Tracker. And so when Neil founded the center, and he tasked us with getting something into patients' hands within 5 years. And that's, for anybody who is familiar with drug development, that sort of rules out, new drugs to kind of like satisfy that, because drugs take a long time to develop. And so we started to think, is there something that we could do where we might better understand the course of inflammatory bowel disease or IBD through the microbiome that would allow us to use the drugs that we already have better. And so, for example, if I could predict that in 3 weeks, you were going to have a flare-up of inflammation in your inflammatory bowel disease, then maybe I could treat you today instead of waiting until you actually had symptoms, and maybe that would be more effective. And so that's the idea behind the IBD Tracker clinical study. We've already enrolled half of the patients at a site in the Netherlands, and we're just starting to enroll patients at MGH. And these patients will, they'll get data from a wearable device. They'll get microbiome samples every week. They'll get to see their microbiome samples, and we'll use all that data to help predict flare-ups in advance. So that's kind of one project I'm really excited about. The other resources I'm quite excited about are a lot of the work we're doing on strain banking. So we generated one of the most comprehensive libraries of microbes that are found in people in the Boston area, which is pretty similar to people anywhere in the world who are living a modern industrial lifestyle. We've as I mentioned, expanded that now through the Global Microbiome Conservancy, and we're starting to preserve biodiversity from indigenous populations. And then we started something called Strain Atlas, where we take one particular species, and then we isolate the same species from hundreds or even thousands of people to look at all the different variants from one person to another. So even if you and I have exactly the same bacteria, it might only share 50% of its genes in common, like your strain versus my strain. And so we don't know what those other 50% of the genes are doing. And so we're trying to get a handle on that.
Right.
And there's a lot of other disease-related ideas that we're looking at. One fun project is cholesterol. We've identified together with Dr. Ramnik Xavier, some bacteria that can basically convert cholesterol into caprostanol, and caprostanol gets excreted from the human body. So that can actually lower cholesterol levels. And we found bacteria through our work with the Global Microbiome Conservancy, groups of bacteria that work together that can make this process even more efficient. And so that's largely what we've been doing on the science side. That's a good cross section through it. But a lot of what we do is it's a community for the next generation of microbiome scientists. So, we're recruiting brilliant postdocs and graduate students and undergraduates at MIT. At one point, I think the Microbiome Club was the largest undergraduate club on campus, and it's certainly one of the most active.
Yeah, I know there's a lot of different sort of conditions that people are thinking about and a lot of potential, but I also know the group has focused a lot on IBD, and that in fact, that was the reason that sort of Neil first contacted the lab. I wonder, Neil, if you could sort of tell people about how that came about for you.
Well, I'd like to take a minute to talk about some of the new direction in IBD research that I'm quite interested in. Can we put up a slide? So this slide represents well, first of all, everybody's microbiome is different, and that's how we're slowly learning about how this ecosystem works. But there's thousands of different bacteria doing lots of different things, and we don't know where to start in looking at what bacteria contributes to IBD. Here's a chart based on data from thousands of patients. The vertical axis are different strains, and the horizontal axis shows whether those strains are overrepresented in IBD, inflammatory bowel disease, or underrepresented. You can see that there's about 5 or 6 up at the top that are overrepresented and about 5 or 6 that are underrepresented. The overrepresented may be contributing to the condition. The underrepresented are equally important and may be bacteria that we need in order to control the condition, but they're missing. These over and underrepresented are now going to be primary targets for our research into understanding their specific functions, what genes are being expressed, and their role in the ecosystem. This should bring us a lot closer to understanding the mechanisms of inflammatory bowel disease.
Thank you for that, Neil. I know you've done a lot of work and really spent a lot of time thinking about those questions.
Yeah, you can tell Neil is so engaged in the science. It's worth pointing out that Neil is actually the person who brought me into microbiome science. I was doing other things. And Neil, I wonder, would it be okay for you to tell people how you first got involved with microbiome and IBD?
Sure. Well, I had a family member with inflammatory bowel disease, and as an MIT engineer, I decided to research it. And but I'm not an expert in the field. And I was dissatisfied by the lack of any understanding in the medical community related to autoimmune diseases. And the treatment consists of, managing symptoms. And I learned of some promising research in Australia, of all places, suggesting the microbiome was involved. And I started to challenge doctors in the Boston area to study it. And eventually it changed their understanding of the microbiome considerably. But while I was talking to them about their research, I found they were all turning to MIT for technology support, for analyzing their data and understanding it. So that was a real turning point for me. And I came back to MIT to help found the Center for Microbiome Informatics and Therapeutics. And although we make so much progress in learning about all this, I'm extremely gratified to see the number of students focused on the field and the thriving ecosystem in the Cambridge area. But, it's a long haul and we're just getting started.
Yeah. And, Neil, your help has really kind of helped us get started in a really robust way. And it's been so gratifying to work with a philanthropist who's so engaged in the outcome and even in the scientific process. And I think what we've done together is lay this foundation. So we built all these tools that we needed to do what we've done for inflammatory bowel disease. But now I think we're really in a position where we can take advantage of that and apply it to other indications like autism, like cancer, like Parkinson's. And so at this point, we're really looking to find new folks to partner with who care deeply about these indications and who want to get engaged. And it's not just at the level of philanthropy. We need people who are excited about maybe the IBD tracker trial and want to help man the phones and handle some of the logistics and there's many ways to get engaged with us.
Guys, I'm going to try to squeeze in one more question. And so I'm going to make it like a Frankenstein question of a couple of related questions. One person asked just what clinical trials are happening. And another person asked, what's the FDA feeling on this science? And I feel like those are sort of related issues that maybe you could speak to.
Yeah. So there's a number of trials happening at MIT. A lot of them are observational trials. The IBD tracker trial is the one that's closest to my heart and that I spend the most time on day to day. But if you go on clinicaltrials.gov and put in microbiome or put in fecal transplant or something like that, you'll see a huge breadth of different trials. So there's a lot going on out there. And the FDA, well, the FDA is, got pretty slow during COVID because, they were mostly focused on that and they're just kind of getting ramped back up. But there's it's interesting, and for anybody who's interested in drug development and especially regulation, this is a type of drug that doesn't really exist yet, and so we're sort of figuring out the regulatory aspects as we go.
Yeah, I think it's going to be really interesting to see sort of how FDA approaches this.
So the FDA, of course, we should be able to develop a lot of kinds of therapies just following the normal FDA protocols. But when you take fecal material from one person and put it in another one, that's a whole new thing that the FDA has no mechanism for. They'd have to develop a whole new program like they do for blood or tissue. And there's no category for this kind of material, and it would take a long time for us to get there.
Regulators have their work cut out for them in this space.
Yeah.
Before I get to a final comment from each of you, I just want to remind everyone that you are going to get a link to the recording of today's discussion. There was a lot going on. I don't think it all went in my ears, so I will probably review some of it. And it'll also include some resources for you, ways to get involved, and you can feel free to share the recording with anyone who might be interested. So with that, Neil, Eric, any last words?
Sure. I envision a world where low-cost microbiome therapies replace drugs in many cases and prevent or cure many chronic conditions. I think in a generation, we'll look back at people eating broad-spectrum antibiotics to be viewed as a terrible historical practice that embarrasses people. And I think new technologies are required to understand the function of the microbiome and to engineer the solutions. And there's no better place suited for this challenge than MIT.
I just want to add, it's been so wonderful working with you, Neil, over the years, and I'm so proud of what we've been able to pull together. And for other folks who want to get involved, there's going to be links, and there's a number of different ways to engage with us. And so I look forward to hearing from a lot of our viewers after the show.
And I can say that there are a ton of really fascinating questions that were asked that we didn't get to all of, and I apologize for not being able to weave them all in, but this was an extremely engaged audience, and I'm sure you will be hearing from folks who are really curious about this work.
I hope so.
So with that, I want to thank both of you for your insights and your expertise and for having this conversation with me. I really appreciate it.
Thank you, Kelly.
Thank you, Kelly.
Thank everyone who was joining us on behalf of myself and the MIT Alumni Association. Thank you for joining us, and have a great night.